If you have asthma, you know that feeling all too well. That tight chest, the shortness of breath, but what's actually going on inside your airways? Well, today, we're going to unpack the science behind one of the biggest culprits, way too much mucus. It's a fascinating story about these tiny little factories inside our lungs that have just gone into complete overdrive.
Okay, let's just jump right in with a really powerful fact from our source material, because this sets the stage for everything. We're not talking about just a little bit of phlegm here. In the absolute worst cases of asthma, the airways can get totally blocked by these mucus plugs. And that is exactly why figuring out where all this stuff comes from is so incredibly important.
And even when it's not fatal, this extra mucus creates huge problems. It's the reason for that persistent cough and having to clear your throat all the time. It physically makes your airways narrower, which of course makes it harder to breathe. And get this, over the long haul, it actually causes your lung function to decline faster than it should. So this isn't just a side effect, it's right at the heart of the disease.
So, the million-dollar question is, where is all this dangerous mucus coming from? Well, to find out, we have to become molecular detectives. We need to zoom in way in on the lining of our airways and find the exact factories that are cranking this stuff out. And here they are. Meet the goblet cell. These little guys are the main mucus factories in our airways. Now, normally, they're the good guys, right? They're part of our defense system, trapping dust and germs. Their main product is something called mucin. And that's the key ingredient that gives mucus its sticky, gel-like texture. But in asthma, something goes terribly wrong with these cells.
Now here's where it gets really interesting, because this is a super important point. The problem isn't just that each individual factory, each goblet cell, is working overtime. No, the body is actually building more factories. This process of multiplication has a name: goblet cell hyperplasia, and it's one of the key things that goes wrong in an asthmatic airway.
So, just how many more factories are we talking about? Well, the research is pretty clear on this. Folks with asthma have, on average, 2.5 times more goblet cells lining their airways compared to people without asthma. I mean, just think about that. That is a massive increase in the capacity to produce mucus.
And this chart shows you exactly what that leads to. It's pretty stark. We're looking at the amount of mucus that's just stored inside the airway lining, waiting to be released. With 2.5 times the cells, the total volume of stored mucus is about 3 times higher than normal. It's basically a giant reservoir of trouble just sitting there.
Okay, so we've identified the factories, and we know they're working in overdrive, and there are too many of them. But what's telling them to do this? Who's the foreman on this out-of-control factory floor? Let's follow the next clue as we dig into the command and control system for all this overproduction.
So who is sending the orders for the goblet cells to replicate like crazy? Well, the answer isn't actually in the airway lining itself. It comes from the body's own immune system.
The masterminds behind this whole operation are a special kind of immune cell called T helper type 2 cells, or just Th2 cells for short. When it comes to allergic asthma, these are the cells that are really running the show. They orchestrate the whole inflammatory response, and one of their main jobs is to shout the order to ramp up mucus production.
And here's the chain of command, laid out nice and simple. It all kicks off when an allergen, like pollen or dust, gets into the airway. That wakes up the Th2 cells. In response, these Th2 cells release a flood of chemical signals called cytokines. And it's those cytokines that deliver the final order to the airway lining. Build more goblet cells, now!
Okay, but cytokines is a pretty broad term. It's like saying messages. To really get what's happening, we need to decode the specific molecular messages that these Th2 cells are sending. What exactly are they saying?
Scientists have narrowed it down to a few key players. These are called interleukins. Specifically, IL-4, IL-9, and IL-13 are the main messengers that directly tell the goblet cells to multiply. Then you've got another one, IL-5, which doesn't talk to the goblet cells directly, but it calls in other inflammatory cells that make the whole situation worse. You can think of them as different managers, all working on the same project: more mucus factories.
So here's the really critical part. How do these cytokine signals actually work? Well, they flip switches at the genetic level. They turn on specific genes inside those goblet cells, and the most important one is called MUC5AC. This gene is literally the recipe for the main gel-forming mucin in our airways. It's the blueprint for the very gunk that clogs up the lungs.
And this data right here just shows you the proof.
You can see that while a few different mucin genes are turned up, that spike from MUC5AC is huge. It confirms that this is the main recipe being used to create the mucus that causes all the problems in asthma. So the cells aren't just multiplying. They're being genetically programmed to churn out this one specific, very troublesome product.
All right, so the factories are built. The product line is running hot. The warehouse is full. But how does all of this stored mucus actually get out of the cells and into the airways? Well, the release mechanism itself is this amazingly fast and honestly kind of explosive process. I absolutely love this description from the source material because it's so perfect. The mucins are packed super tightly inside these little packets called granules. When the cell gets a signal to release, the granule merges with the cell wall. Water instantly rushes in, the mucin molecule expands like crazy, and it erupts out of the cell just like a jack-in-the-box, getting way bigger in a split second.
But hold on, how do those granules even get to the cell wall to be released? Well, recent discoveries have pointed to a critical protein called MARCs. You can think of MARCs as a kind of molecular crane or maybe a logistics manager inside the cell. Its entire job is to move those mucus packets into position so they can be launched.
And here's the play-by-play of how it works. The cell gets a signal that says, "Release the mucus." That activates MARCs. It detaches from the edge of the cell, moves into the cell's main body, and physically hooks onto a mucus granule. Then, using the cell's internal skeleton like a system of ropes and pulleys, it literally drags that granule to the surface, ready for that explosive jack-in-the-box release. It's an incredible physical process. So, what's the point of all this? Why map out this entire super complex pathway from the immune cell signals to the genetic recipes all the way down to the molecular cranes? Because every single step we uncover is a potential weak point, a target we can aim for with new medicines. By knowing this entire chain of command, we can start to think up new strategies. What if we could block those Th2 cytokine signals from ever reaching the goblet cells? Or what if we could interfere with the MARCs protein so the molecular crane never delivers its payload? Every single one of these is a brand new possibility for a future treatment.
And this is what makes the research so urgent. The reality is, right now, we have no specific drugs designed to stop this final step, this release of mucus granules, what scientists call degranulation. Creating a treatment that could do that would be a complete game changer for people with asthma and other airway diseases.
And that really brings us to our final thought for today. As we get better and better at understanding all these tiny, intricate details, it raises this amazing possibility. Could a future treatment for one of the worst parts of asthma be as targeted and as precise as simply telling one single protein, like MARCs, to just stand down? That's the hope that's driving all this science forward. Thanks for joining us.